Students, welcome you back to my channel Biomix. Hope you're doing well. So have you ever wondered how a single phytohormone can control multiple factors, like it can be formation of shoot, it can be growth of the leaves, or it can be delaying the aging of the plants. So these are being controlled by a single phytohormone which is known as cytokinin. So in this today's video we will discuss about the signaling pathway of cytokinin. So before discussing about that, we have to first of all understand where is the cytokinin formed. So the cytokinin, it is being synthesized in the root and it is being transferred to different parts in the acropetal manner, and it is by the xylem. The transfer is by the xylem. So this signaling pathways of this cytokinin, it is being derived from a bacterial two-component system. So in the prokaryotic two-component system, it is having a sensor protein. In the sensor protein, there is a histidine kinase which is present. So in this protein, there is an input domain and a transmitter domain. The other protein which is present in the prokaryotic two-component system is the response regulator protein. So this sensor protein, it sends, and in the response regulator protein, it gives the response after the sense is being taken by the input domain. So after a sense is being taken by the input domain, if something is given to the input domain, there is phosphorylation of the histidine domain, and this histidine domain, it is a kinase. So now this transmitter domain which is having a histidine with it, it is being phosphorylated. After the phosphorylation, it is being activated. So as this was a kinase, so it can transfer a phosphate to the other part. It will transfer the phosphate to the receiver domain. This receiver domain, it is having a asparatate with it. So after this asparatate is being phosphorylated, this output domain will help in giving the response, and the genes will be expressed over here. Okay, this is how the prokaryotic two-component system is working. From this, the cytokinin signaling pathway is being derived. Like we will see how it is being derived. So in the cytokinin signaling pathway, there are other multiple steps which are present apart from the general prokaryotic two-component system. So here we can see there is a hybrid sensor histidine kinase domain. So these are the receptors of cytokinin. Like the receptors of cytokinin can be CRE1, AHK2, AHK2 or AHK1. Okay, so here there are three types of domain. Like it is having a input domain, it is having a transmitter domain, and it is having a receiver domain. Okay, so this is known as hybrid sensor histidine kinase. Okay, next is the histidine phosphotransfer domain. So this is responsible for transferring the phosphate to the receiver domain. It takes from this receiver domain. So here there is a histidine sensor, hybrid sensor histidine kinase. So from here, after getting the input from the cytokinin, in presence of cytokinin, there is autophosphorylation of this histidine, and this is a kinase present over here. So from this histidine, there is transfer of this phosphate to the asparatate which is present in the receiver domain. Okay, so this phosphate is transferred again to the HPT domain which is carrying the histidine. Okay, so this histidine this is working as a histidine phosphotransferase. So the phosphate which is present, it will be transferred to the receiver domain. So in case of cytokinin, the example of the HPT protein is AHP1. Okay, so now this HPT domain which is carrying the phosphate, it is being transferred. This phosphate is being transferred to the receiver domain, and this receiver domain is having a asparatate with it, and this domain is of the response regulator. Okay, in case of the cytokinin, this example of this response regulators are type B transcription factor and ARRs. After getting this phosphate from this HPT domain, and the output domain which is present over here, it gives signal or it responses to this gene. Okay, or we can tell that it responses to this phytohormone, and it gives and it is helping in the expression of the genes. Now we will discuss, this was how the prokaryotic 2 system, from the prokaryotic 2 component system, how the cytokinin signaling pathway is being derived. Now we will understand how the mechanism of the signaling pathway of the cytokinin is taking place. So in case of cytokinin, it is having a receptor present. So this receptor, it is a 2 component receptor. Okay, so here the receptor is it can be CRE1, AHK2 or AHK3. So there is a portion where the cytokinin can come and bind. So this has been binded in the chase domain of this receptor. Okay, so this receptor is having one chase domain, a histidine kinase domain and a receiver domain. This is the receptor. Okay, so here, by utilizing the ATP, this phosphate from this histidine, after a cytokinin is present over here, there is autophosphorylation of this histidine and it is being transferred to the asparatate. Okay, so after this after the phosphorylation of this asparatate, what is this? It help in the phosphorylation of this histidine, which was a transmembrane domain. We can see here histidine it is a histidine phosphotransferase. So here the histidine phosphotransferase is AHP. Okay, so this AHPs which is present, it can transfer inside the nucleus and it can phosphorylate the receiver domain, and this receiver domain is having a asparatate over it. Okay, so here there is phosphorylation of this receiver domain, due to which there is a connection of this output domain. So here there will be synthesis of some other genes. So this AHP which is present, it can also phosphorylate type A ARRs. So it is also having a asparatate with it. So here this AHP which is carrying the phosphate, it can transfer this phosphate to this asparatate of type A protein. Okay, so when this type A protein is being phosphorylated, it will negatively regulate this by inhibiting this AHP. Okay, so what happens when this receiver domain, this is the type B, this is type A ARRs and this is type B ARR. So when this receiver domain get this phosphate, there will be some other genes which can be formed. So here we can see the type B ARRs which is present. Now they will come and bind in the promoter region of this DNA. So due to this binding of this type B ARRs in this DNA, there will be synthesis of this type A ARRs. Okay, and there will be formation of the mRNA of the ARRs and there will be synthesis of the ARRs. Okay, now due to this there will be a cytokinin response. So this type B ARRs which are present, it is being regulated by 26S proteasome. So here the type B ARRs which are present, it is ubiquitinated by the SCF KMD. KMD is the kiss me domain. It is the kiss me deadly protein. It is a kiss me deadly protein which is present, and it is a F box protein. It is a F box protein and it is being degraded by 26S proteasome. In this way it is being regulated. So what happens if there is degradation? If there is degradation of this type B protein, there cannot be synthesis of this type A ARRs. So there will be less cytokine response. Okay. So what happens next? The various genes which are whose transcript level are altered by type B ARRs as well as the downstream target mediate the response of the cell to cytokinin. So in this way the cytokinin response is taking place when the type B ARRs are being synthesized. This is how the regulation of the cytokinin signaling is taking place. If the cytokinin responsive genes are being synthesized, so what they will do, they will help in the formation of the shoots, they will help in the growth of the leaves, as well as they will delay the aging of the plants. In this way the cytokinin is regulating the plant. So I hope this videos will be helpful for you, and if you are having some doubt regarding this lecture, please please ask me in this comment section. So